Hello, welcome back to video 3 in the urinary system series. I am really excited because in this video we're going to start to really get into the weeds of urine formation. Very exciting. In the previous videos we went through the anatomy, went through the functions of the urinary system. It'll be really helpful for you to know the anatomy, especially the anatomy of the nephron, as we begin to talk about how these structures work together to filter the blood and form urine. Oh God, I'm so excited. But again, when am I not? Okay, so we're going to break down urine formation into 3 steps. Let's see if I can figure this out again. Oh, there we go. Okay. This is a new program, so bear with me. Keep it with black. All right. So we're going to be using these 3 steps. Urine is formed through first glomerular filtration, and then tubular reabsorption and secretion. And the third and final step of urine formation is water conservation. And these happen in order. We may remember that all of this is happening inside of the nephron. So think back, we talked about the renal cortex, the outer rim of the kidneys, which is chock full of nephrons. And it is in these nephrons that we filter the blood and form urine. In addition to forming urine, we can't forget that other function of the urinary system. Because the kidneys are able to secrete more or less of substances and fluid in the urine, this process is huge in homeostasis. So the kidneys have the ability to excrete more or less fluid through urine. And in that way, it can maintain homeostasis of blood volume. And because the kidneys filter the blood and form urine, and because they can excrete more or less substances, they can help to maintain homeostasis of solutes. For example, think about glucose. If we have too much glucose in the bloodstream, one way to get rid of it is for the kidneys to excrete more glucose in urine. So we can't forget the goal, the added function of maintaining homeostasis. Here are some numbers for you. Maybe not memorize the exact numbers, but the idea that the kidneys filter a lot, and I mean a lot of blood each day. So in a 24-hour span, the kidneys may filter between 150 and 180 liters of blood. And out of that 150 to 180 liters of blood, only about 1 liter, maybe even 1 to 2 liters, is what's urinated out as waste. So what does that mean? That means that 148 to 178 liters of fluid is reabsorbed. So the kidneys filter so much, and because the body likes to recycle and reuse what it can, the kidneys are able to reabsorb a lot and reuse it for the body. And we only pee out under normal circumstances a very small amount. All right. We're going to come back to this one, but we're going to just jump right in to step 1. How do I get, there we go. Okay. In step 1, we have glomerular filtration. And we already talked about this because we know that the nephron has 2 parts. The first part of the nephron is the renal corpuscle, which has the glomerular blood vessels surrounded by Bowman's capsule, which then leads into the tubules, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and then collecting duct, right? Glomerular filtration is happening in the glomerulus. We're filtering blood. But I want to talk about how specifically we're filtering the blood. Let's see if I can switch colors here. Oh, that's the wrong one. Bear with me. Let's talk about some red. Actually, yes, do red. So if we were to look at the glomerulus, remember the glomerulus is a ball of capillaries. And if we look at the glomerular membrane, We may remember that the glomerular membrane has in between it those podocytes with little feet-like extensions that wrap around the capillaries, which form filtration slits in between. And it is between those filtration slits that we filter. All right, so imagine these are the filtration slits. All right, excellent. I'm going to go back to black here. I think I have to sneeze, so if I sneeze, I apologize. Excuse me. So how the filtration happens is based off of size. And what I mean is, I think of the glomerular filtration kind of like running things through a strainer or a colander in the kitchen. So if you make some pasta and you just bring it out of the boiling water, you pour the water and the pasta through a colander or strainer, and that colander has little holes in it, much like these filtration slits. And what happens is whatever is small enough to fit through those spaces diffuses out or filters out. On the other hand, whatever is large, it's just simply too big to fit through the filtration slits. And that's exactly how glomerular filtration works, just like a strainer. The blood is filtered by size. Whatever is small, and it gives you here the exact size, but you don't have to know the exact size. Whatever is small is filtered out of the glomerulus and into the tubules. More specifically, it's the proximal convoluted tubule. But whatever is small enough can fit through the filtration slits and moves into the tubules. Whatever is large is not able to be filtered out of the glomerulus, and instead, the large particles leave the glomerulus and they leave through the efferent arterioles into the peritubular capillaries. Or if it is a juxtamedullary nephron, it would be into the vasa recta. Now I know I'm throwing a lot at you, and at the end we're going to bring it all together. Hold on for a sec, I've got to blow my nose, I'm so sorry. Allergies don't care if I'm giving a lecture or not. So we're going to talk more about this later and put it all together, okay? But if you can just stay with me each step of the way, make notes, understand what I'm talking about. Okay, so the first step of urine formation is glomerular filtration. It happens in the glomerulus. The blood comes in and is filtered. Whatever is small enough is filtered out of the glomerulus and moves into the tubules. Versus whatever is large is too big to fit through the filtration slits, and instead leaves the glomerulus. It's not filtered out into the tubules, but it leaves the glomerulus, the large particles, via the efferent arteriole and out into the peritubular capillaries. So what's interesting is that we're filtering by size, strictly filtering by size. The blood filtration here is not based off of what's good and what the body can reabsorb versus what's bad and has to be excreted. So we're not, the glomerulus isn't going off of that at all. It's strictly by size. So we're not really worrying about what's a waste product or what's something the body wants to reabsorb. That's not the job of the glomerulus. It's pretty much strictly filtering by size. What we're going to find out is that the tubules, so the next step of urine formation, it is the tubules that have the job of deciphering what's good and useful to the body versus what is a waste product. Pretty cool. I mean, I think so. All right, I'm going to go back for 1 second because I want to talk about something that we already know, but we got to make sure we talk about it. In order to filter, we must have two things. We talked about that glomerular membrane that has these filtration slits, right? anytime we try and filter, we must have at least a semi-permeable membrane. Indeed, our filtration slits, it's semi-permeable, so we can check that off the list. And we also, in order to filter, have to have some kind of pressure. In other words, there's got to be some kind of pressure that's going to help drive the small molecules through the filtration slits. And indeed, we do have some pressures, which we're going to talk about next. All right? Okay. I'm going to skip over to exactly those pressures. All right. Let's go here. Perfect. These pressures are going to sound familiar because we already talked about them. And that's the benefit of A&P2. Hopefully, each time we go through it, it's going to settle into your brain more and more. We rely on opposing forces. And we know these names. We have a hydrostatic pressure and we have osmotic pressure. Now, it gets a little bit more tricky here in the kidney. Let's just start with the basics because these should sound familiar. And this is a great picture for us to demonstrate that. So I'm going to switch to the color of white. So here is the glomerulus, inner capillaries. Here's the afferent arteriole. So blood is going to enter the glomerulus through the afferent arteriole. Whatever is small enough is going to be filtered through the filtration slits into the capsular space and into the tubules. Whatever is too large, if something is too large and it can't fit through the filtration slits, it will stay in the glomerulus for a period of time and eventually leave the glomerulus via the efferent arteriole. But let's go back because I want to talk about the hydrostatic pressure. Let's do this one in green. All right, so we're going to show hydrostatic pressure in green. Hydrostatic pressure is really similar to blood pressure. We think about blood pressure. Hydrostatic pressure is the pressure that the blood comes into the glomerulus with. And this BHP, blood hydrostatic pressure is going to be a pressure that goes in the direction of inside the glomerulus to outside the glomerulus into the capsular space. On the other hand, we have an opposing osmotic pressure, and that's what these blue arrows are showing you. This is colloid osmotic pressure. This is pressure from the capsular space in to the glomerulus. So we're always going to have some of both. The hydrostatic pressure is helping to filter fluid out of the glomerulus into the capsular space, and the osmotic pressure is the resistance force from the capsular space back in to the glomerulus. Don't worry about capsular hydrostatic pressure now. What I want to talk about is the net. So what is the net? The net, here we go. Still learning. What did I use for the hydrostatic pressure? I don't even remember. We'll use white, I suppose. The hydrostatic pressure goes out, and the osmotic pressure comes in. Which is greater? Well, as you can see here, we want the hydrostatic pressure to be greater than the osmotic. And you can see that there's about a net filtration pressure of 10 millimeters of mercury. So let's put some numbers on it just to give an idea here. So as you can see at the bottom, the hydrostatic pressure, oh, well, you can't see it when I'm doing it in white now, am I? The hydrostatic pressure is about 60 millimeters of mercury. So if I go back here, the hydrostatic pressure coming in here, it's about 60 millimeters of mercury. So if the hydrostatic pressure is 60 millimeters of mercury, then we have the opposing osmotic pressure. And actually, we didn't talk about the capsular pressure, which is fine. We're just going to put them together. So together, these two make up about 50 millimeters of mercury. So the osmotic pressure is about 50 millimeters of mercury. These are the exact conditions we want. We want the hydrostatic pressure to be greater. And what that means is we've got a balance always pushing fluid this way. This is really important. If we don't have this balance, then we're going to lose filtration, right? Because what happens, let's see. Guess you can't erase it after you already do it. Oh, there we go. Okay. What happens if the fluid coming in, what if the fluid coming in becomes greater? Then we're going to lose the ability to filter blood out. And basically, the kidney function will cease. But we'll talk about that more later. We just want to know that the hydrostatic pressure should be greater than the osmotic pressure. And that's what keeps the kidneys be able to filter blood. All right, I'm going to come back to glomerular filtration rate. That's a regulation topic, so we're going to come back to that. I know you're so sad that I'm skipping through all of these, aren't you? That'll be in our fourth video. Okay. Hopefully you didn't have any alcohol, or else I'm going to make you dizzy here. All right. So let's talk about the second two steps of urine formation. We just talked about the first two, or the first step, and that was glomerular filtration. We filter the blood in the glomerulus. Whatever was small enough continues into the tubules. Whatever was too big stays in the glomerulus and then leaves into the efferent arteriole into the tubules. So now we have small things here in the tubules, and we have large things here in the surrounding capillaries. But we haven't yet figured out what we want to reabsorb and keep in the body and what we want to excrete as urine. And that is exactly the job of step number 2 of urine formation: tubular reabsorption and tubular secretion. Off we go. Let's first talk about tubular reabsorption. Let's see, I wish I had this picture down here again, but that's okay. You know why? Because I can draw it. So we have the tubules, and then running right alongside the tubules are the peritubular capillaries. Right? This is the same issue we have here, the same picture we have here. We have the tubules in yellow and the surrounding capillaries in red. Tubular reabsorption, tubular secretion tend to happen kind of concurrently. Let's first talk about tubular reabsorption. This can be tricky, but as long as you know the entire definition, you're going to be good. Tubular reabsorption is reabsorption of good things from the tubules back into the bloodstream. So remember, previously we had small things here and large things here, but we don't yet know what's good and what's waste. So in tubular reabsorption, we reabsorb good things, things that the body can use. We reabsorb good things from the tubules back into the bloodstream. So if there's any particles in the tubules that our body wants to keep, we reabsorb them from the tubules back into the bloodstream. Because whatever is in the bloodstream stays in the body. Whatever is left in the tubules eventually goes on to become urine. You can see a listing here of some things that tend to be reabsorbed. You don't have to know them specifically, just be able to define tubular reabsorption. What you should know, however, is that most of the tubular reabsorption happens earlier in the tubules, mostly in the proximal convoluted tubule and the loop of Henle or nephron loop. There's much less reabsorption happening in the distal convoluted tubule. And of course, I mentioned this already, we reabsorb a lot. I'm not going to ask you that number, but just know that we tend to reabsorb a lot and leave very little left for urine. So, all right, here's a picture that gives us some more examples. I'm going to talk more about this later, so don't worry about this picture yet. We're going to come back to that. I did give you some tables here, so if you're curious about what specific substances are reabsorbed where, it's possible to break it down because there's specific solutes that are reabsorbed in the proximal convoluted tubule versus the loop of Henle, descending and ascending limb. And there are some different processes. Sometimes it's active transport, sometimes it's diffusion. It just depends, but you don't have to know any of these. Oh, there's even more. Look at that. But you don't have to know them. So on the PowerPoint, if you have the PowerPoint, I think this is animated, or I know the ones are later. But again, it's important to know for tubular reabsorption that we're moving good things, what the body can use, and make sure you know the direction. We are moving from the tubules back into the bloodstream. From the tubules back into the bloodstream. Okay? Good. And here's an example of some mechanisms. You don't have to know them. I mentioned some of them, but you don't have to know them. I want to talk about tubular secretion. So let's set up our same thing as we had before. So we have the tubules, right? And then alongside it, we have the peritubular capillaries, okay? And if you remember, in tubular reabsorption, we moved good things. So I'm going to put reabsorption, we put good things from the tubules back into the bloodstream. So the black arrows are tubular reabsorption. Apparently I can't spell at the same time as I'm thinking. But now let's talk about tubular secretion. So at the same time, now we're talking about secretion of waste products from the bloodstream back into the tubules. So now we're talking about waste that's in the blood, and now we're secreting it from the blood back into the tubules. This is tubular secretion. So it can be a little tricky, and the words of reabsorption and secretion don't tell you whether it's a good product or a waste product, and they don't tell you which direction things are going. So that's why it's important to know. In tubular secretion, we have waste products that are moving from the bloodstream into the tubules. Think about it. We don't want waste to stay in the bloodstream. So if, after the initial glomerular filtration, if there is some waste products left in the blood, we don't want to keep the waste products in the blood, so we secrete them into the tubules, because whatever's in the tubules goes on to become urine. You should know that this is happening mostly later on in the tubules. in the distal convoluted tubule, whereas tubular reabsorption happens more in the proximal convoluted tubule and loop of Henle. We tend to have more tubular secretion in the distal convoluted tubule. Oh, I didn't know we could zoom in. We're in this together with this new app. We're gonna get it. All right. So here again, I've tried to do some pictures just so you know. Whereas, let's see. In this picture, I'm using white and black. So white would be tubular reabsorption, where we move good things from the tubules back into the bloodstream. But when we talk about tubular secretion, we're moving waste products from the bloodstream back into the tubules. Okay, so our first step of urine formation was glomerular filtration, where we filter out by size. Our second two steps were, well, it's one, but they're both happening at the same time. Tubular reabsorption, tubular secretion happening in the tubules. This is where we put waste products into the tubules so they can be excreted as urine. And this is where we keep good things into the bloodstream to be reabsorbed back into the body. The third and final step of urine formation is water conservation. I'm going to keep this pretty simple. Water conservation is water is reabsorbed from the ducts, from the collecting ducts, back into the bloodstream. Because the first step happened in the glomerulus, the second step happened in the tubules, and the final step happens in the collecting ducts. Before we excrete urine, the body wants to reabsorb and keep as much water as it can. So water conservation simply means in the collecting ducts, a lot of water is reabsorbed from the tubules back into the blood. We want to keep as much fluid as we can. The body likes it, it needs fluid. We only want to have as little urine as possible with water. Okay. I've given you some review things here. You can study any way that you can. All right. I've got a couple slides here on drugs acting on the kidney. You do not have to know any of this for the exam. I added this because people ask me so many questions about it. And a lot of medications, particularly medications that affect fluid volume and blood volume, act on the kidney. So you can see loop diuretics. An example of a loop diuretic is Lasix. A diuretic makes you pee more, right? So Lasix works in the loop of Henle. That's why it's in the category of a loop diuretic. So cool. You can read all about this. This shows you where in the loop of Henle that medication is going to work. Again, you don't have to know any of these, but just extra information. There's a different classification called thiazide diuretics, but you don't need to know these. I got a little carried away, but okay. Last thing I want to do for this part is I just want to give you some examples of how different products make their way through the kidneys. I'm not going to ask you this on the exam, so if you want to, you can just fast forward. But I think it is really interesting because it helps us understand the steps. So if you remember, we did glomerular filtration based on size. So that just meant that small particles went into the tubules and large particles went into the bloodstream. Well, sometimes a small particle is a waste product. So that means that the initial filtration by size of the glomerulus is all we need to do. Because it was small, it went into the tubules. And if it's identified as a waste product, we have nothing else to do but to keep it in the tubules to be excreted as urine. But other times, that small product is actually something that we can use. It's good. So if that small product is something good, we actually have to reabsorb it. So there's different scenarios, and I give you some examples of these. Okay. So insulin, for example. Insulin is something that is relatively small, so it's filtered out of the glomerulus into the tubules. But once insulin has its action in the body, it's a waste product. So it's a small product that's filtered out and is already a waste product, so nothing else has to happen. It is released as urine. Boom. But talk about glucose. Sometimes glucose is relatively small, in this case, so it can be filtered out of the glomerulus into the tubules. But unless it's excess glucose, if you're a non-diabetic, if we get glucose here in the tubules, our body wants to reabsorb it. So we actually, even though it was filtered out, it's in the wrong place. So we have to reabsorb it from the tubules back into the bloodstream. Again, that would not be true of a diabetic. Let's talk about this bottom one, drugs. So let's give an example. Again, this is not going to be on the test, but I think it can be helpful if you want to just practice understanding. So what if we had something that was too big? Remember those large products? So we can have some drug metabolites, some medication metabolites, and they tend to be big. And remember, things are filtered by size in the glomerulus. So something that's too big to be filtered out of the glomerulus is actually going to stay in the glomerulus and then leave via the efferent arteriole into the capillaries. But a drug metabolite, we don't want that. So here we have something that's large, but it's actually a waste product. So even though it stayed in the bloodstream, we actually have to secrete it back here. So there's different scenarios. Not going to be on the test, but different scenarios. Okay, let's make this the last topic. Let me just look ahead. Yeah, let's make this the last topic. Then we'll review, and then our last video will be talking about regulation. Okay, there's something called the countercurrent. You might see it referred to as the countercurrent multiplier. And what this refers to is reabsorption in the loop of Henle. The loop of Henle has the ability to reabsorb what it needs to in order to maintain homeostasis. Let me actually just see what the next slide has to say. Yeah, this can get pretty tricky, so let's try and simplify it. If I was going to define the countercurrent, how can we define it simply? I would probably say ability of loop of Henle. That's where it happens mostly, or you could say nephron loop. Ability of loop of Henle to reabsorb more or less to maintain homeostasis. That's going to be our simplified definition. If I was going to simply define the countercurrent, the ability of the loop of Henle to reabsorb substances, reabsorb either more or less of them, in order to help maintain homeostasis. It's called countercurrent because it does involve an osmotic gradient, but I don't think that's something we need to get into. Let's just get the basics. So let's talk about it more. And what I want to say is a couple things. Stick with me because you're going to get it. Let's first address in the descending limb. It is in the descending limb of the loop of Henle where there is primarily water absorption. So we want to memorize in the descending limb, it's more about water reabsorption. Versus in the ascending limb of the loop of Henle, it's more about sodium reabsorption. And this picture shows us that. In the ascending limb, it's more about sodium reabsorption. This is just memorization. The descending limb does more of water reabsorption, and the ascending limb does more of sodium reabsorption. Okay. The last thing to talk about with this topic is how water reabsorption and salt reabsorption affects osmolarity. There's that word again. Oh, here I am having problems. Just want to have, there it is. Our good old friend osmolarity. Let's use green. We may remember that osmolarity, that's bright, isn't it? Osmolarity is, this kind of hurts my eyes. I'm not going to be using much green in the future. Concentration of the solute. Whew, wonder if I can change the color. Osmolarity, concentration of the solute. How about green? Okay, darker green. So if we have a high osmolarity, we have a lot of solute. I give the example in A&P1 and in A&P2 of Kool-Aid. A high osmolarity Kool-Aid has a lot of sugar in it. So it means that the fluid is very dense. Versus a low osmolarity solution has low amounts of solutes. In the example of Kool-Aid, if you don't have a lot of sugar in the Kool-Aid, you have more water, it tends to be more watery. Look at what happens to the osmolarity as water is reabsorbed and then as salt is reabsorbed. These numbers here are referring to the osmolarity. It starts off going into the descending loop of Henle at about 300. I think it's milliequivalents or something like that. The unit doesn't matter. Oh, milliosmoles, there it is. But what I want to point out is, during the descending loop, as water is reabsorbed, the osmolarity goes up. We're removing water, so the relative solute concentration inside the tubules goes up. Think of the Kool-Aid. If I have Kool-Aid running through the tubules, and I remove water, but I keep the same amount of sugar in it, it's going to become a sweeter, more dense. And you can see the color gets deeper orange. So in the descending loop, the osmolarity goes up. We're reabsorbing water out of the tubules, which makes the relative solute concentration go up. But as we turn the corner and come up to the ascending limb of loop of Henle, now we're reabsorbing solutes, and the osmolarity goes back down, because now we're reabsorbing, in the Kool-Aid example, reabsorbing more sugar, which makes the osmolarity go down. This is what the countercurrent multiplier is. The ability of the loop of Henle to reabsorb more or less water and solutes helps to maintain homeostasis of these solutes. In the process of doing so, the osmolarity of the fluid will change, but that's okay. And this shows you those osmolarity numbers going down into the medulla, but don't worry about that. All right, so I'm going to go through these last couple slides, then we'll end this video and we'll save the fourth and final video for regulation and some odds and ends. If you have the PowerPoint of this, it is animated, so you click the mouse or the arrow key and it goes through the different ones. Here it doesn't let me do that, but that's okay. So I'm trying to put it all together because we covered a lot. If we take a more global view, we may remember that blood is going to come in to the kidneys through the renal artery, then go through the segmental artery, into the interlobar artery, to the arcuate artery, to the interlobular artery. Then we switch gears to this magnified view on the right. Here was the arcuate artery up close, interlobular artery. Branching off of the interlobular artery is the afferent arteriole into the glomerulus. And what happens in the glomerulus? There's filtration. Whatever was small gets filtered out of the glomerulus into the tubules. Whatever's in the tubules is now called filtrate. Whatever was too big leaves the glomerulus via the efferent arteriole into the peritubular capillaries. All right? Okay. Then, step 2, we have tubular secretion and tubular reabsorption. Now we have small products in the tubules and large products in the bloodstream. But sometimes we see a need to do reabsorption or secretion. Reabsorption, tubular reabsorption, is the movement of good things from the tubules back into the bloodstream. Tubular secretion is movement of waste products from the bloodstream back into the tubules. And this is happening all along the proximal convoluted tubule, the loop of Henle, and the and the distal convoluted tubule. In the end, when it's all said and done, sorry about that. Whatever is left in the tubules goes into the collecting duct. We might have some additional water reabsorption. That's the third step of urine formation. Whatever's left in the tubules continues on through the minor calyx, major calyx, renal pelvis, out the ureter to the bladder, and then out the urethra as urine. Versus whatever's left in the bloodstream eventually makes its way back into the interlobular vein, arcuate vein, interlobar vein, renal vein, wait for it, inferior vena cava, back to the heart, right atrium, right, and it can be recirculated. Okay, I'm going to end there. In our 4th video, hopefully our final one, we're going to come back and talk about regulation. Good job, folks.